Hi everyone, and welcome to Logical Pharmacology. Ever wondered how your body just knows what to do? Like when your heart starts racing before a big presentation? Or your pupils get bigger when you walk into a dark room? That's your autonomic nervous system doing its thing. But before we get into how it pulls that off, let's take a quick step back and look at the nervous system as a whole. Now, the human nervous system is huge. We're talking over 86 billion neurons in the brain alone, plus billions more spread throughout the body. And these neurons, they're all linked by a massive number of connections, more than 100 trillion synapses, constantly relaying signals to keep things running smoothly. A typical neuron has a cell body. You might hear it called the soma or perikaryon, just to keep things interesting. Branching out from that are a bunch of short extensions called dendrites. They are like the antennae, picking up incoming signals. Inside the cell body, you've got all the gear that keeps the neuron alive and firing. A big nucleus, mitochondria, Golgi apparatus, and the rough endoplasmic reticulum. At one end, there's a little bump called the axon hillock. That's where the axon begins, a long cable that carries the message away from the cell body. Now, axons come in all sorts of lengths. Some are just a millimeter long, others can stretch over a meter. And at the end of that axon are tiny terminals called boutons, little buttons packed with chemicals that help the neuron connect and talk to its next target, usually another neuron or a muscle. This is called synaptic transmission. But neurons aren't just sending messages, they're constantly receiving them too. Most of the input comes in through the dendrites or the cell body. But fun fact, some signals can even be picked up at the axon terminals. All this communication happens at the synapse, that tiny gap where one neuron hands off information to the next. And depending on where that handoff happens, you get different types of synapses. Axodendritic, where the axon of one neuron connects to the dendrite of another. Axosomatic, where the axon links up directly to the soma or cell body. And axoaxonic, where one axon connects straight onto another axon. Neuron receives input either from other neurons, or in the case of sensory neurons, directly from the environment. Typically, the flow of information goes like this: dendrites, cell body, down the axon, terminal boutons, then to the another neuron. Now, when we talk about a synapse, we're talking about three key parts. 1. The axon terminal of the neuron sending signals, we call it presynaptic neuron. 2. The synaptic cleft, the tiny space between neurons. 3. And the postsynaptic membrane of the receiving cell. Here's a cool twist. In the autonomic nervous system, the neurons innervating target organs don't just stop at one little bouton like they usually do. Instead, they've got varicosities, about 20,000 little swellings spread out along the length of the axon. These release neurotransmitters not at one point, but along the way in what's called a synapse en passant. This setup lets them bathe the whole target tissue in neurotransmitters, activating a large area all at once. It's like throwing a bucket of paint instead of using a paintbrush, perfect for widespread control of smooth muscle, glands, and all the other things the autonomic system looks after. Let's look at how we classify neurons. First up, according to the number of processes arising from the cell body, we've got the pseudo-unipolar neuron. This one has a round cell body with a single process coming out that quickly splits into two long branches, one heading out to the body called the peripheral process, and the other going in toward the spinal cord called the central process. The branch that travels toward the organ is part of a peripheral nerve, and it picks up sensory input from places like the skin, muscles, or even the gut. At the far end of this branch, it has terminal endings that either act as sensors themselves or connect to receptors that do the sensing. The other branch travels into the spinal cord and carries that information into the central nervous system. Even though it splits in two, the whole thing functions like a single long axon. Next, we've got bipolar neurons. These are simpler in shape. They've got one process at each end of the cell body, an axon and a dendrite. You'll see these in more specialized systems, like the retina or the inner ear. And finally, multipolar neurons. These are the most common type. More than 99% of all neurons in the body fall into this category. They've got lots of dendrites sticking out from the cell body and a single thin axon that sends signals out. Most motor neurons are multipolar. Neurons can also be grouped by type of input and the direction of flow of information. The afferent neurons, also called sensory neurons, carry information from the body towards the brain and spinal cord. Think of things like touch, pressure, pain, taste, even gut stretch. All those signals get passed into the central nervous system to be processed. Efferent neurons, or motor neurons, do the opposite. They carry information away from the central nervous system to muscles and glands to cause a response. That could be contracting a muscle, increasing heart rate, or releasing a hormone. We also classify neurons by the kind of chemical messengers or neurotransmitters they use. For example, if a neuron uses glutamate, it's called glutamatergic. If it releases a peptide, we call it peptidergic. Simple, right? Now let's zoom out and look at the nervous system as a whole. It's divided into two main parts, the central nervous system, or CNS, which includes the brain and spinal cord, and the peripheral nervous system, or PNS, everything outside the brain and spinal cord. The peripheral nervous system connects the central nervous system to the rest of the body. The peripheral system has two subdivisions. One, the somatic nervous system, which handles voluntary movements, things like moving your arms or walking. Two, the autonomic nervous system, which manages automatic, SAM, unconscious body functions like heartbeat, digestion, and breathing. The somatic nervous system uses two types of fibers. Somatic afferents bring sensory information in from things like your eyes, ears, nose, tongue, skin, and joints. Somatic efferents send motor signals out to skeletal muscles so you can perform voluntary movements. So when you raise your hand or wiggle your toes, that's your somatic system in action. The autonomic nervous system, or ANS, also has afferent and efferent fibers. Autonomic afferents bring in sensory info from your internal organs. These include pH receptors, stretch receptors, pain sensors, chemoreceptors, all the things that monitor what's happening inside. Autonomic efferents send signals back out to smooth muscles, cardiac muscles, glands, and even the enteric nervous system in your gut. Now here's where it gets interesting. When we talk about the sympathetic and parasympathetic nervous systems, we are specifically referring to the visceral efferent fibers, the ones that control the output to internal organs. The visceral afferent fibers, the ones carrying signals into the brain, technically belong to the autonomic system too. But because they don't neatly fit into the sympathetic or parasympathetic divisions, some experts prefer to group them under the broader term visceral nervous system instead. And then there's the third part of the autonomic nervous system, the enteric nervous system, often called the second brain. It's embedded entirely in the wall of the digestive tract and controls digestion from start to finish. Now, the sympathetic and parasympathetic systems can influence it, but the enteric nervous system can run independently, even if it gets cut off from the brain. The autonomic nervous system helps your body respond to internal changes and external stress. It works quietly alongside the somatic system, sometimes even before it. One of the standout features of autonomic nervous system is how fast and intense its effects can be. Within seconds, it can double your heart rate and blood pressure, trigger a full body sweat, empty your bladder, or crank up your gut activity. Autonomic nervous system is always running in the background, fine-tuning your body whether you're resting or active. The autonomic nervous system innervates all organs and tissue in the body, except skeletal muscles. Skeletal muscles are handled by the somatic nervous system. So the autonomic nervous system is made of three parts: sympathetic, the fight or flight system, activating the body in times of stress; parasympathetic, the rest and digest system, calming things down and conserving energy; enteric, the gut's very own nervous system, which exclusively controls digestion. All right, so now we've got a handle on the big picture of the autonomic nervous system. We'll focus on the sympathetic and parasympathetic nervous system here, and save the enteric system for a future video. But first, how does autonomic information actually travel? It goes both ways, from the periphery to the brain through the afferent pathway, from the brain back to the periphery through the efferent pathway. Let's start with the afferent pathway. The afferent pathway sends sensations from your internal organs, their attachments, or the membranes covering them, straight to the central nervous system. These signals help trigger automatic responses without you needing to think about them. Now, these sensations can be conscious, like when your stomach feels bloated after a big meal, or the dull pain you feel during reduced blood flow, what we call ischemia. Unconscious, like changes in blood, pH, osmotic pressure, or even the temperature inside your head. You're not aware of these signals, but your body is. The neurons that carry these signals are called visceral afferents, and depending on their location, they come in three main types: vagal, spinal, and enteric. Let's break them down. Vagal afferents gather sensory signals, things like blood pressure and stretch, from areas such as the carotid artery and the aortic arch. The information travels through their peripheral process to a cell body sitting in the nodose ganglion. From there, it continues along the central process of the vagus nerve into the brain stem. Spinal afferents work in a very similar way. They collect information from thoracic, abdominal, and pelvic organs, and then carry it into the spinal cord. Their cell bodies sit in the dorsal root ganglia just outside the spinal cord. Both vagal and spinal afferents are pseudo-unipolar neurons. They've got one long process that splits in two, one branch reaching out to the organ and the other heading into the central nervous system. Then we've got the enteric nervous system, basically the nervous system of the gut, and it works a bit differently. Inside the gut wall, there are four types of sensory afferents. Two of them are extrinsic. One follows the spinal nerves, like we just covered. One follows the vagus nerve. The other two are truly intrinsic. They never send signals to the brain or spinal cord. Intestinofugal neurons send signals out of the gut wall to nearby sympathetic ganglia, triggering local reflexes there. Intrinsic primary afferent neurons, IPANs, stay within the gut wall, connecting to other enteric neurons to coordinate reflexes right there. No brain required. Even though the sensory endings of vagal and spinal afferents are embedded in the gut wall, they're still considered extrinsic because their cell bodies sit outside the gut in nodose or dorsal root ganglia. Pretty clever system, right? The gut sorts a lot of things out on its own. Now, about those nerve endings, they're spread out over a wide area in each target organ, with plenty of varicosities scattered near specialized cells that release chemicals like ATP to trigger the afferents. Take the bladder, for example. When it stretches, the urothelial cells stretch too and release ATP. That activates nearby sensory endings, and that signal shoots up to the brain. Once these signals reach the spinal cord, they enter through the dorsal roots, usually into lamina 1 or 5 of the dorsal horn. Vagal afferents, on the other hand, send their info to the nucleus tractus solitarius in the lower brainstem. From both of these points, information is sent on to higher brain centers, like the hypothalamus, which regulates autonomic responses, and the ventromedial thalamus, which processes stimuli and alertness. The medulla oblongata also plays a key role, helping to coordinate these reflexes. All this sensory input doesn't just affect autonomic functions, it also influences emotional and even immune responses. So, visceral afferents are incredibly important. They detect changes using free nerve endings, and they transmit signals with vesicles loaded with serotonin and other neuropeptides. Before we move on, let's quickly define a few terms you'll hear often. In the central nervous system, that's the brain and spinal cord, groups of nerve cell bodies are called nuclei. If they are laid out in layers, we call them laminae. If they're in columns, they're called columns. Bundles of axons here are called tracts or fasciculi. And a group of tracts is called a funiculus. Out in the peripheral nervous system, everything outside the brain and spinal cord. Groups of cell bodies are called ganglia. Axons run in bundles called fibers. Multiple fibers plus connective tissue and blood vessels make up nerves. Nerves can merge to form rami or roots. And a plexus just means a branching, interwoven network of nerves. Same word we use for blood vessels. The efferent pathway. Let's break down how signals travel from your brain or spinal cord to the organs they control. This is what we call the efferent pathway in the autonomic nervous system. It's a two-neuron relay system, a preganglionic or a presynaptic neuron, and a postganglionic or a postsynaptic neuron. The preganglionic neuron has its cell body in the central nervous system, either the brain or spinal cord, and it sends its axon out to a ganglion in the periphery, where it meets the postganglionic neuron. This is where the information is passed, specifically the neurotransmitter. In the sympathetic system, the preganglionic fibers are small, myelinated, so signals travel quickly, and they release acetylcholine, which is why we call them cholinergic. In the sympathetic system, these preganglionic neurons live in the spinal cord. Their axons shoot out and connect with postganglionic neurons in one of the autonomic ganglia. Now, the postganglionic neurons are a bit different. Their axons are unmyelinated, and they end in special branches called varicosities. These are like little swellings that spread out across the target organ. Usually, these postganglionic fibers release norepinephrine, making them adrenergic. But there are some exceptions. For example, in the sweat glands and some blood vessels in skeletal muscle, the postganglionic fibers still release acetylcholine. Over in the parasympathetic nervous system, the setup is pretty similar but with a few key differences. The preganglionic neurons come from two areas, the cranial region, that's the brain, and the sacral region of the spinal cord. Just like in the sympathetic system, they are myelinated and cholinergic. They release acetylcholine. But instead of synapsing in big ganglia far away from their target, parasympathetic neurons synapse in ganglia that are often right near or even inside the organ they're controlling. That means the postganglionic fibers in this system are usually shorter than their sympathetic counterparts. And just so you've got the full picture, in the somatic nervous system, it's even simpler. There's just one single neuron, also cholinergic, that connects directly to a muscle cell. No relay, no ganglion, no fuss. All right, now let's locate where these pathways actually start inside the body. Here's a spinal cord showing the significant cranial regions and spinal regions. For the sympathetic nervous system, the preganglionic neurons live in the spinal cord, specifically between the T1 and L2 or L3 levels. That's why we call it the thoracolumbar outflow. For the parasympathetic system, the preganglionic neurons come from the cranial region in the brain, the cranial outflow, and the sacral region between S2 and S4 in the spinal cord, the sacral outflow. That's your craniosacral outflow. If you're curious where exactly in the brain these parasympathetic pathways begin, here's a quick look one. Edinger-Westphal nucleus in the midbrain. Remember? Nucleus is a group of cell bodies in the central nervous system. So from this, a large number of preganglionic neurons arise, whose axons or fibers follow the cranial nerve 3, oculomotor, 2, superior salivatory nucleus in the pons, kicks off cranial nerve 7, facial, 3, inferior salivatory nucleus, that's where cranial nerve 9, glossopharyngeal starts, 4. And finally, nucleus ambiguus and dorsal motor nucleus of the medulla. These give us cranial nerve 10, the famous vagus nerve. All right, now let's zoom in on the spinal cord and see where the sympathetic and parasympathetic pathways actually kick off. The spinal cord has that classic layout. White matter on the outside, gray matter on the inside. The gray matter split into 3 main regions. The dorsal horn at the back, the posterior of our body. The ventral horn at the front, anterior of our body. And the lateral horn tucked in the middle. Now that lateral horn, also called the intermediolateral column, only sticks out in the thoracic and upper lumbar segments, from about T1 down to L2 or L3. And this is the exact spot where the preganglionic neurons of the sympathetic system start. From each level of the spinal cord, 2 roots come out on either side. The dorsal root at the back carries sensory input into the spinal cord. Those fibers belong to pseudounipolar sensory neurons with their cell bodies sitting in the dorsal root ganglion. The ventral root at the front is the motor root, sending signals from the spinal cord out to muscles, glands, and organs. The dorsal and ventral roots join together to form a spinal nerve, and that's why each spinal nerve has both sensory and motor fibers bundled together. Pretty efficient. All right, so the spinal nerve splits into 2 main branches, the dorsal ramas and the ventral ramas. The dorsal ramas handles the muscles and skin on your back, sending motor signals out to control them and bringing sensory info back to the central nervous system. Meanwhile, the ventral ramas takes care of the front and sides of your body in the same way. If you look at the spinal cord full picture, I've illustrated the cervical and lumbar segments above and below the thoracic section. You'll notice something interesting. The lateral horn is missing from both the cervical region and the lumbar region below L2. Now, those preganglionic neurons we talked about, they've got to pass the baton by synapsing with postganglionic neurons. But where does that happen? At a ganglion, right? Well, running down either side of your spine, from the neck all the way to your lower back, you'll find pairs of these ganglia. They're not just hanging out solo, they're connected to each other above and below their own level, forming a chain called the Prevertebral Ganglia, or more simply, the sympathetic chain ganglia. Think of them as relay stations linking up all the levels of your spinal cord, so the sympathetic nervous system can send quick, coordinated messages all over your body. Each spinal nerve links up with the sympathetic chain through 2 small branches, the white ramas communicans and the gray ramas communicans. The white ramas carries preganglionic fibers, and the gray ramas carries postganglionic fibers. We'll dive deeper into those in just a moment. You can notice that other than the lateral horn, there's something else that's absent in the cervical spinal cord. And below lumbar segment 2, the white ramas communicans. Now the preganglionic neurons originate in the lateral horn. From there, they travel through the ventral root and then the white ramus to a ganglion at the same level within the sympathetic chain. At the ganglia, the preganglionic neuron meets the postganglionic neuron. Once this synapse happens, The postganglionic fibers head back to the spinal nerve through a gray ray mass communicans. Through the spinal nerve, these neurons go to the target organs. Remember, these ganglia are present at each level of the spinal cord and communicate with each other up and down the spinal cord. But have you noticed? White rami are absent above T1 and below L2. This means only those pairs of sympathetic chain ganglia from T1 to L2, which have white rami, will receive inputs via the white rami. So, how do the ganglia in the chain above T1 and below L2 get their preganglionic fibers for synapsing? Here's how it works. The ganglia above T1 get their input from fibers coming from thoracic segments, T1 to T5. These fibers travel upwards, climbing the sympathetic chain, before synapsing in those higher ganglia. The ganglia below L2 get their input from fibers coming from T5 to L2. These fibers travel downwards in the chain, descending from the lower thoracic and lumbar ganglia before they synapse. Why is it white and gray ray mass communicants? Preganglionic neurons are myelinated, and they travel in the white ray mass communicants, which is why it looks white. Postganglionic neurons are unmyelinated, so when they leave through the gray ray mass communicants, it appears gray. Remember, white rami communicants are only found between T1 and L2 because only these segments have preganglionic fibers leaving directly to the chain. Gray rami communicants are found at every spinal level because every spinal nerve can carry postganglionic fibers to their targets. So even though preganglionic neurons only originate between T1 and L2, regions above and below still receive their sympathetic supply. The fibers simply travel up or down the chain from other ganglia before synapsing, and then send their postganglionic fibers back out through gray rami at that level. On both sides of your body, the sympathetic chain stretches all the way from the base of the skull down to the lower end of the vertebral column. Right at the bottom, the two chains meet at a single unpaired ganglion called the ganglion impar. This trunk runs the entire length of the vertebral column, and it's clever. It lets sympathetic fibers that originate only from a small section of the spinal cord, T1 to L2, be distributed all over the body. Next, we have prevertebral ganglia, which you might also hear called collateral ganglia, preaortic ganglia, or subdiaphragmatic ganglia. These are unpaired autonomic ganglia in the abdomen and pelvis. So, not all sympathetic fibers do their business inside the sympathetic chain ganglia. Some take a different route. The preganglionic fibers that arise from spinal segments below the diaphragm pass through the paravertebral sympathetic chain without synapsing there. They also skip the gray rami altogether. These preganglionic neurons also travel up or down the sympathetic chain, but they do so without synapsing in the paravertebral chain ganglia along the way. Then they exit the sympathetic chain, joined with similar fibers from other spinal levels, forming a splanchnic nerve that carries them to a prevertebral ganglion. The main prevertebral ganglia include celiac, superior mesenteric, and inferior mesenteric ganglia. Now, the prevertebral ganglia are where the preganglionic fibers finally synapse with postganglionic neurons. From there, the postganglionic fibers form small visceral nerves that spread out to the abdominal and pelvic organs, usually as part of a plexus, interwoven and branching network of nerves. The main prevertebral ganglia include celiac, superior mesenteric, and inferior mesenteric ganglia. So in short, preganglionic fibers from T5 to L2 skip the chain, travel in splanchnic nerves, synapse in prevertebral ganglia. Then the postganglionic fibers travel to organs via arterial pathways. Note, not all splanchnic nerves terminate in prevertebral ganglia, while thoracic splanchnic nerves, greater, lesser, and least, synapse in prevertebral ganglia like the celiac ganglia. There are other types of splanchnic nerves, like the lumbar splanchnic nerve, that bypass the paravertebral chain and also contribute to the formation of the pre-aortic plexus, with some fibers continuing onward without synapsing in any ganglia. So this is an exception to the two-neuron chain of the sympathetic nervous system. The adrenal medulla. For the adrenal medulla, the preganglionic sympathetic fibers travel through the splanchnic nerves and pass through the prevertebral ganglia, like the celiac ganglion, without synapsing. These fibers then directly terminate on the chromaffin cells of the adrenal medulla. This is why chromaffin cells are said to function as a modified postsynaptic neuron, releasing neurotransmitters into the bloodstream rather than onto a specific effector cell in an organ. Alright, now that we have covered the chain ganglia and the prevertebral ganglia, let's quickly summarize the different ways sympathetic fibers leave the spinal cord and reach their targets. You can think of it as 5 main pathways. 1. Branches to spinal nerves. These are the fibers that re-enter the spinal nerve through the gray Rami communicants, the basic route, unlike the white Rami, which only exists from T1 to L2. The gray Rami are found at every spinal level, one for each of the 31 pairs of spinal nerves. These postganglionic sympathetic fibers then hitch a ride with the cutaneous branches of the spinal nerves to reach their targets in the skin. That means sweat glands, erector pili muscles, the little muscles that make your hair stand up, and the smooth muscle in peripheral blood vessels. 2. Branches to cranial nerves. Some sympathetic fibers connect to cranial nerves, often cranial nerves 9, 10, and 12. They might join these nerves directly, or they might follow blood vessels until they're close enough to hop on. This is how they reach their target structures in the head and neck. These are actually output from superior cervical ganglion. To summarize the route, preganglionic sympathetic fibers originate from the spinal cord, primarily at spinal cord segments T1 to T4. These fibers then ascend within the sympathetic trunk, reaching the superior cervical ganglion. It is at the superior cervical ganglion, the uppermost ganglion of the sympathetic chain, that the preganglionic fibers synapse with postganglionic neurons. The postganglionic fibers from the superior cervical ganglion then exit the ganglion and travel via various pathways, including forming the carotid plexus and traveling with cranial nerves to innervate structures in the head and neck. 3. Branches that wrap around arteries. Many postganglionic fibers simply wrap themselves around blood vessels, such as the internal and external carotid arteries or the vertebral artery. These arteries act like highways, carrying the fibers along to reach specific target tissues. And it's not just these major arteries. Plenty of smaller vessels carry sympathetic fibers too. 4. Direct branches to specific organs. Some postganglionic fibers take a more direct route to certain organs, like the eyes or the heart, without having to join a spinal or cranial nerve first. They might still travel alongside arteries or in a nerve bundle for part of the journey, but they mostly go it alone. Remember, these are not splanchnic, they are postganglionic, meaning they have already synapsed before leaving the sympathetic chain. 5. Branches to autonomic plexuses. Sympathetic fibers also head to large autonomic plexuses, networks of mixed sympathetic and parasympathetic fibers. Carotid plexus receives sympathetic fibers that travel alongside the carotid artery to supply structures in the head and neck. Cardiac plexus contains sympathetic nerves that originate from the cervical and thoracic ganglia, which then innervate the heart. Pulmonary plexus. Sympathetic fibers also extend to the lungs as part of the pulmonary plexus. Celiac, superior mesenteric, and inferior mesenteric plexuses are prevertebral or pre-aortic plexuses surrounding major abdominal arteries and contain a large number of sympathetic neurons. They receive input from thoracic and lumbar splanchnic nerves and innervate the abdominal organs and their associated arteries. Renal plexus have got sympathetic nerves from the thoracic and lumbar splanchnic nerves. These synapse within the renal plexus to innervate the kidneys and regulate renal blood flow and function. Superior and inferior hypogastric plexuses are located in the pelvis. They receive sympathetic fibers from the splanchnic nerves, including the lumbar splanchnic nerves, and are involved in the innervation of pelvic organs. Here's the trick. Fibers going to the heart, lungs, and pelvic organs are usually post-ganglionic. Fibers going to the abdominal plexuses, like the celiac, superior mesenteric, and inferior mesenteric plexuses, are usually pre-ganglionic, and they'll synapse in ganglia within the plexus or even in the wall of the target organ. So, between these 5 roots, sympathetic fibers can spread from a relatively small section of the spinal cord, T1 to L2, to just about every corner of the body. Alright, now let's jump into the parasympathetic nervous system, starting with the cranial part, which kicks off with 4 key cranial nerves. First up is the oculomotor nerve, 3. Its cell bodies hang out in the Edinger-Westphal nucleus. The preganglionic fibers run through the oculomotor nerve all the way to the eye's orbit, where they synapse with postganglionic neurons in the ciliary ganglion. From there, the postganglionic fibers hop onto short ciliary nerves, piercing into the eyeball to control the sphincter pupillae muscle, which makes your pupils constrict, and the ciliary body muscle, which helps adjust the lens so you can focus on things up close. Next, the facial nerve, 7. Its cell bodies live in the lacrimal and superior salivatory nuclei. The lacrimal nucleus sends axons to the pterygopalatine ganglion, where they synapse with postganglionic cells. These fibers then work on the lacrimal gland to produce tears, and the nasal mucosa glands to promote mucus production. Meanwhile, axons from the superior salivatory nucleus reach the submandibular ganglion to synapse with postganglionic neurons, which then stimulate saliva flow from the submandibular and sublingual glands. Then we have the glossopharyngeal nerve, 9. Its cell bodies sit in the inferior salivatory nucleus. Preganglionic fibers head to the aortic ganglion, where they meet postganglionic neurons. These postganglionic fibers leave the ganglion to innervate the parotid gland, triggering saliva flow, and some minor salivary glands in the oral mucosa, helping keep your mouth nice and moist. Finally, the big player, the vagus nerve. Its fibers come from the nucleus ambiguus and the dorsal motor nucleus. Preganglionic parasympathetic fibers from the nucleus ambiguus connect with postganglionic neurons in the cardiac ganglia around the great vessels of the heart. These postganglionic fibers then innervate the sinoatrial SA node and sometimes the atrial and ventricular muscle fibers, all helping to slow down your heartbeat. The vagus nerve also sends out plenty of visceral branches along the way. These branches contribute to nerve plexuses related to thoracic organs and a large prevertebral plexus in the abdomen and pelvis. These plexuses often mix in sympathetic fibers too. When parasympathetic fibers are present in these plexuses, their postganglionic neurons hang out in the walls of the target organs themselves, sacral portion of the parasympathetic nervous system. The preganglionic fibers from cell bodies in the spinal cord segments S2 to S4 jump onto their matching spinal nerves and make their way to the pelvic plexus. From there, they connect with small scattered ganglia close to the pelvic organs, where they synapse with both the enteric nervous system and the postganglionic parasympathetic neurons. This setup is kind of similar to the sympathetic system, where preganglionic fibers also travel along spinal nerves to synapse in ganglia. But the key difference? In the sacral parasympathetic system, These ganglia are right near, or actually inside, the target organs, giving it a more local, hands-on role compared to the sympathetic chain ganglia. So that's your logical walkthrough of the autonomic nervous system. In the next video, we'll dive deeper into the innervation and functions of both the sympathetic and parasympathetic systems, all explained logically. Don't forget to like, share, and subscribe to Logical Pharmacology. We logify the tricky stuff so you don't have to memorize it.